Okay, I think we're ready to rock. Thank you everyone that's able to attend. This is a kind of a culmination of a couple of years worth of work, where we really geared this meeting towards the farmers that have been collaborating with us. And we've generated and put together some reports for the specific fields that we were working with them on. And so this is geared towards kind of providing a little bit of feedback and back to the farmers, and for them allowing us to work on their fields. And we were collaborating with about 20 farmers across the state. And in addition to those farmers that are joining this meeting, we have our other collaborators, such as our collaborators with MFA, Corteva, different agronomists and research individuals that we've been working with during the course of this project. And we're grateful that everyone was able to come and hopefully have a good discussion about some of the results and the trends that we're seeing across the state regarding the interaction of soil health and these soil health tests and traditional soil fertility. I'd like to thank the advisors that have been able to help Matt and I. My personal advisors are Newell Kitchen, Steve Anderson, and Kristen Viam. And then Rob Myers also serves on my committee. They've been a great help in guiding this project and helping us understand and discern the data that we're seeing. And then also thank especially Corteva. They've provided some significant funds in helping this project work and get up off the ground and go. So a lot of this is because of their efforts in helping us. And then we, through the University Extension, and through MFA, they connected us with a lot of the different farmers that we're collaborating with. So we'd like to say thank you to all those that have supported this project so far. Just a general overview of the meeting. First, we're going to have Newell Kitchen give a kind of a broad overview of how this project and how this question of questions of interactions between soil health and soil fertility, how this project came to be. And after that, Matt Henry, the other graduate student that's working on this project, he's going to give an overview of the farmer reports that we have given to the farmers, kind of breaking them down so that we're able to digest them. I'll be posting a link, I believe, in the chat. For those that don't have reports or that aren't a farmer, you can go to Google Drive, and I will post a demo report that you'll be able to look at so that you have something to look at while you're doing it. Or you can just watch the presentation that Matt gives. He breaks it down pretty well. After that, we're going to go through a couple of case studies across the state that we've seen so that we can help interpret these tests. Then we're going to have a break. And during the break, we're going to play a video that we produced. And then after that break, we're going to go into and really dive into the results. Matt's going to first discuss the tissue results from the tissue results across the state, and then I will be diving into the yield responses, and then also some of these different management practices influence on these soil health tests. So that's kind of an idea of what we're going to see today and what we're going to discuss. If you have to leave early, that's totally we understand. So hopefully feel free to ask questions, type them into the chat box as they come. After at the break, we're going to be fielding questions, and then after the results section, after we discuss our results, we will be able to discuss and answer questions there. So with that, I am going to turn the time over to Newell to give a brief overview of this project and kind of see a big picture before we dive in.
Thanks, Jeff. And I will mention it with the questions. Why don't you put them in just as a general so everybody sees the questions, because sometimes someone else's question will prompt you to ask a question. So everybody can kind of see the questions are being asked, and we'll, rather than send them to Matt or Jeff specifically, send it to the kind of a general. So I'm going to start off. Hopefully the screen is showing now, okay? And talk a little bit about where this project started. And it's important to understand that there's kind of a little bit of a paradox that has kind of risen, and we're trying to reconcile that with this project. And it starts with first kind of understanding this big question of what is soil health and how does it relate to soil fertility. And that's starting probably 8-10 years ago. I went to a lot of meetings. Some of those were producer meetings, and they were asking this question of what is soil health, and does this make a difference when it comes to soil fertility? And really, to answer that question, we really have to understand what do people mean when they say soil health. If we put 10 people who've been exposed to some aspect of soil health and you ask them what soil health is, you're likely to come up with 10 different definitions. So I kind of want to put this in the context and kind of help everyone else see at least how I look at soil health. And to do that, I'm going to start with talking about it from the standpoint of the soil chemistry and soil fertility. And actually, there's 3 major portions of soil science that I want to talk about. So this first one is soil chemistry and soil fertility. And this is where I got my formal training, where I spent much of my career, is in this soil fertility nutrient management area. And there's a whole bunch of different measurements in that are taken to kind of properties that are used to help us understand soil chemistry or the fertility as it relates to a plant. So that's kind of one camp. And then you've got another area, and that's the soil physics, the physical properties. And there's a whole bunch of additional properties and indicators, I guess, that we would use to describe soil physics. And that's out there. And it's been there for years. And it's our understanding of soils from that dimension has really grown over the years. And then you have also soil microbiology, and how it looks at a soil, how we characterize a soil with different properties of soil microbiology. So I like to think of this, and this analogy is probably used too much, but we talk about a 3-legged stool. And when it comes to kind of the main different components of soil science, you have these 3 different things that are working together to help us to understand the different functions of a soil. And that's what this overlaid over the top of this now. We have 5 different areas there that we think about in terms of soil and how it functions. And all of these properties, the chemical, the physical, the microbiological, all are working together to kind of help us understand how this soil functions. And when it's functioning in ways that are optimal for the different type of soil that you have, then that's often referred to as soil health. It's a good, healthy soil. So soil health it's kind of hard to put your arms around, just because depending on what part of this diagram you're sitting on, and that's kind of your favorite area, you kind of want to talk about it from that perspective. And so soil health is very broad, and just like, again, it's been compared to like human health, but there's so many aspects of this soil health, and it's important to recognize that's a big, for many, it represents a lot of things when it comes to soil and soil function. Now, there's another definition of soil health that is that's kind of a working definition that a lot of people have started using, and that is the soil health is kind of just fallen into describing the microbiology or the properties associated with the soil microbiology. And so sometimes some people will say soil health and they're just referring to this, and sometimes they're referring to the big picture. So I think it's really helpful to have a context of sometimes people mean different things when they say soil health. So with these different ways of looking at soil health, we have these 2 different camps. And I grabbed this off of the University of Minnesota Extension website to represent one side of the camp. And here it says, can soil health tests determine fertilizer needs? This is posted on their website. As farmers increasingly use soil health tests, now in this case, they're talking about the the biology type tests, many wonder if these test results can or should determine fertilizer needs or for corn and other Minnesota crops. These tests, such as the Haney test, help measure soil health indicators such as the I can even speak this, microbial activity, the carbon, the amount of carbon in the soil, and nutrient availability. While these tests can be useful for contrasting management practices, they shouldn't be used to determine fertilizer application need or rates. So they're coming right out and they're saying you shouldn't be using this. So that's kind of this one camp. These are individuals that have perhaps really are grounded in soil fertilizer decision making based upon the standard soil fertility testing that's been around for about 7 decades. Okay, so that's the one side. And then you've got another camp that says you'll see headlines that are kind of like this: healthy soils need less fertilizer, or something equivalent to that. And so that's another different perspective. And so this is where there's a little bit of some conflict going on, because these multiple voices are out there and everybody's a little confused by, well, how am I to reconcile this? How am I to bring understanding to how I should be making decisions on my field when I hear this person say this and another person say this, or I see this in a documentary and I see this in another documentary. So many hundreds of people are asking this question: what is the relationship of soil health with fertilizer applications? How do we get to a better understanding of what we do? And that question was the thing that started this research. But the approach that we took from the beginning is not to abandon what was in place already. And what has been in place for, like I said, about 7, 8 decades, or maybe even a little bit longer, has been the soil chemistry approach to understanding what fertility, what fertilizer recommendations need to be made. And as you look at this chart, you can see there's the chemistry is the main part of this, and there's a little touch maybe to soil texture, there may be a touch to organic matter, but for the most part, the soil microbiology and the soil physics are not really addressed at all as a part of fertilizer decisions. And that's the culture we've been in, and there are millions of acres that the decisions of what kind of fertilizer should be applied to the fields are based upon those standardized soil tests that have been calibrated to thousands and thousands of field plot research, and then that's the basis of the fertility. But we don't want to abandon that. So the question that we've kind of brought to this project was, can standardized soil fertility tests be improved? It's not to throw it away, but can it be improved by including some soil biology measurements or some additional physical tests? Can the standards be improved if we were to bring those in and bring those into the standard recommendations that we currently have? So that's where we're going to go, and that's the story you're going to hear today with these presentations. And I'll end with so this project has been very ambitious, and it's taken 2 very ambitious individuals to do this. The 2 graduate students, Jeff Svedin and Matt Henry. Jeff is working on his PhD in the soil science department here at University of Missouri, or it's in the natural resources, School of Natural Resources. And Matt Henry is doing a master's degree in the plant science agronomy program here at the university. And so the 2 of them have been working together on this, and they've been great to work with, because they caught the vision of this and they took off, and they've worked with many of you that are on this meeting today. And as Jeff indicated, your cooperation has really helped to make this happen. So that's the setup for what we're trying to find out. The big question: can the standard soil fertility tests be improved upon by using some of these other tests, or are the current tests alone as good as we're going to get? With that, I think I'm going to turn this over to Matt Henry, and he's going to share his screen and take it from there.
All righty.
Just while Matt's getting ready, Newell, I think there might be a farmer that needs permission to get in or something. Christopher Hudson was trying to get in and I think he was asking for a permission code or something.
The permission code. Okay.
Oh, he just said it's working now, so we're good.
Okay. All right.
So thank you, Noel, for our introduction. So with all that in mind, I'm gonna go ahead and jump into these farmer reports that we sent out. And I'm also gonna go over some of the soil health analyses and what they show and you know what we did to actually do those. So I'm gonna get started with this farmer report. So yesterday, all of our farmers should have received reports coming from me, hopefully, that you know, each of you got your own reports for your own fields. So I'm just going to go through a basic outline here and tell you what each thing in the report actually means and how to interpret it. So this is just a picture of the front and the back of the report. So we'll jump straight in. So each report has a picture of the field. So each, you know, you got like 3 or 4 different reports depending on how many fields we had with you. So each report starts with that field's picture. And in that field picture, we also have little pins in there. So each pin represents a stamp. So in this field, we had 3 stamps at the top and 2 stamps at the bottom of the field. And then moving on from that, we have these graphs. So what do these graphs actually mean? So within these graphs, there's points, there's different colored points. So I'm going to try to explain to you what each point means. So in this example, I have the blue diamond labeled 1 with an arrow pointed at it. So that point is where stamp 1 reads on this graph. So in this example, stamp 1 has a reading of about 110 pounds per acre of phosphorus. And then, so that corresponds with where that stamp was in the field. And stamp 3 was up there as well, up there around 100 pounds per acre. So then each of those corresponds with each stamp in the field. And then we have this orange dot. So what's this orange dot mean? This orange dot is the average of the entire field. So in this example, the entire field had not the average of the stamps in that field. So the average of our stamps in this field was about 55 pounds per acre of phosphorus. And then we'll get on to the points or the different parts of this box plot. So the plot, that line that I'm pointing at represents the middle of our data. So out of all of our points, the middle lies right there where that black middle line is. And then if you go up to the top of where the actual box is, that's like the 50% to 75%. And then this would be the 25 to 50%. So if you're within this box, you're within the middle 50% of our data.
So just to make sure it's clear, this is all fields, all producers, the whole study, right, Matt? Okay.
Yep.
All right.
And I think this actual box plot that I'm showing here is from last year, but it's updated for this year's. It means the same, you know, the stuff's the same. You know, what each thing means. So then up here you have your 75 to the top. So if you're up here, you know, you're really high compared to the rest of the stuff in our data set. And then if you're down here, you're pretty low compared to the rest of our data. So then you may be wondering what this red line is. So the red line represents the critical value that is established in Missouri. So for phosphorus, it's, I believe, 44, 45 pounds per acre. So as you can see, over 50% of our data is actually below that critical value. So, we try to target low phosphorus areas while also keeping in mind, we have high phosphorus areas too, to try to show that difference when you add fertilizer to the treatments. So this is just on that front page, the top graph or the top figure has all these different graphs in it. So it has your phosphorus levels, your potassium levels, your sulfur levels, and then your CEC and your pH. So obviously there's critical values for the 3 fertilizer or the 3 nutrients, but there's no critical value for CEC or pH, so that's missing. So you can see each of those for each field. And then on the bottom, you have your soil health analysis. So this is your organic matter, and then a bunch of different tests that we ran. So we ran active carbon, soil respiration, total protein, sulfatase, and on this is last year's, we had glucosidase on there, but this year we included phosphatase instead, just because we aren't done with glucosidase yet, because we're running that in the USDA lab. So before I get to the back, I wanted to explain what a response is and how we came to a decision to make it so we show these responses. So I'm going to give this example of calculating one response. So in this is just an example. These aren't actual numbers. So in this field, for example, I better start with what we actually did. So this picture here is a stamp. So in each stamp, we have 4 different treatments. So we have a controlled treatment, which it says no fertilizer, but it actually got some nitrogen. And then we have potassium, phosphorus, and sulfur, respectively. So in this example, I'm just using the potassium treatment. So when we're calculating a response for this potassium treatment, what we do is we take the yield out of this treatment and we divide it by the yield out of this control. So what that's going to do is tell us how the corn responded to that added potassium. So if you had a treatment 2 yield of 220 bushels per acre and the treatment 1, the control, only had 200 bushels, you would divide those and get a ratio of 1.1, which is also a 10% response. So that's where these responses come into play. And the tissue is, instead of bushels per acre, for the tissue, what we did was we did the concentration of that potassium in the plant, or phosphorus or sulfur, and we just divided that by that concentration out of the control as well. So the graphs on the back show these responses for your individual field. So this is just an example. So you have your different stamps. So 1, 2, 3, and 4, those correspond with the stamps on that front page. And so we'll do stamp 1, for example. So stamp 1 didn't have a response to added potassium, but it did have almost a 10% response to added phosphorus. And then it didn't have a response to sulfur. And so on this, how high the bar goes is basically how high of a response that you saw in that field or in that stamp. And then each color represents the different nutrients. So that's how you read those, and the yield is the same. So yeah, you interpret these the same way. They're just, the yield response is how many bushels per acre, in comparison, whereas the tissue is the concentration of that nutrient in the plant. So I believe that's everything out of the reports. So now I'm going to move on to the different soil health analyses that we did and are shown in those reports. And I'm going to tell you about each one and what they represent, basically. So the different tests that are shown on the reports are organic matter, active carbon, soil respiration, total protein, and then 2 enzymes. We have sulfatase and phosphatase. So starting with organic matter, I hope everyone probably knows what organic matter is, I'm assuming, because it's talked about all the time. So what is organic matter? Organic matter is a storage of soil carbon. It slowly releases plant available nutrients, and it acts as a long-term carbon sink. So organic matter doesn't break down super fast, so it can act as a long-term sink for your carbon. And generally, higher organic matter means lower farm inputs. So you hear this a lot, where if you have a higher organic matter, your soil is inherently going to be more productive, which isn't always necessarily the case, but that's generally what they say. And this organic matter has a strong influence to your water holding capacity in the soil. So I have this figure here. So your organic matter is a measure of how well you can store carbon, and there's different carbon pools that are included in this, which some of our other tests are going to help show, just this active carbon pool, for example. But there's different carbon pools that do different things, and that is all included in this organic matter test. So if we move on to active carbon, so like I said, there's different carbon pools. So active carbon is a measure of that active pool of the organic matter. So this test is a test of the readily available carbon out of that organic matter, and this measures the food and energy source for the microbial community in the soil. And this picture here is an example of different levels of this test. So this test, you use a dark purple material, or a dark purple compound, and based off how much active carbon there is, it changes the color, and we read this color to determine the amount of active carbon in the soil. So this test has been able to detect differences in management practices, such as tillage or no tillage, or cover crops or stuff like that. So this is a pretty good test to, gauge where you're at in your soil health. So moving on, next we have soil respiration. So soil respiration is a short-term carbon mineralization shown by amount of carbon dioxide released in a 4-day incubation period. So this is a very broad measure of your microbial activity, which estimates metabolic activity. So basically what we do here is we add water to the soil, and when we add that water to the soil, the microbes are going to become active, and they release carbon dioxide. And when they release, based off how much carbon dioxide is released, we can measure that, which gives us an idea of the total amount of microbes in the soil. So this is a pretty broad measurement, and it's still in that active pool of carbon. So this graph here, it's basically comparing that active carbon and soil respiration measurements, and if one's higher versus the other one being higher. So down here on the bottom, this mineralizable C, this is the soil respiration, and then this is your active carbon. So what this is telling us is if you have a high soil respiration value and a low active carbon, so you're going to be over here on the graph, basically what that means is that you're not building up your organic matter. It's basically using what you could be building up as organic matter. Instead, it's using it as minerals for your crops. Or if you had a high active carbon, so if you're up here on the graph, if you have high active carbon and low soil respiration, then you're building up organic matter. So the goal is to be out here where you have high of both, so that way you can, be building organic matter, but also using it as minerals for your crops that you're growing. So a couple more, we have total protein. So this is your readily available organic nitrogen that can be recycled and then taken up by plants. So it's a measure of the size of the pool of nitrogen being converted to that plant available form. So basically it serves as a reservoir of nitrogen that, will become available in that next growing season. So this can be a very helpful test to show how much nitrogen that is in your soil that will become available. And then finally, we have the two enzymes. So I group these together because, you know, enzymes, they perform pretty much the same role. They're just for different nutrients. So these are responsible for converting organic sulfur and phosphorus to plant available forms. And I also have on here that some enzyme activity has been shown to depend on the amount of available nutrients. And for example, sulfatase activity depends on the amount of sulfur supplied to the microbes. So what I'm trying to say here is if you have a lot of sulfur that you're putting down as fertilizer, there's going to be a lot of sulfur available to those microbes. So in return for that, they don't produce a lot of sulfatase. So it's like a negative feedback to where if you add sulfur, there's less sulfatase. If there's not a lot of sulfur available, there's more sulfatase and stuff like that. So I think with that, I am finished explaining the report. And I think Jeff said he wanted to wait to do questions until after he was done with his. So if you have any questions, you can write them in the chat and then we will go over them after Jeff's next presentation. So with that, I'll transfer over to Jeff.
Thanks, Matt. So for me personally, and thank you, Matt, for explaining all these different tests. For me personally, I always seem to learn best when I apply the different things that I'm introduced to. And these soil health tests are really things that people are new. You don't hear about you're just starting to hear about these tests. They're not very common. So to really understand, I think, these tests and what they mean on your farms, I'd like to do a couple of case studies, because when I first started looking at these, they were just numbers on a page. So I didn't start to really understand, or they didn't really mean a whole lot to me until I started looking at your different farms. And so I thought that going through a couple of case studies throughout the state might help us understand, or help you understand how these different tests can inform like your practices, or what your management practices are doing, such as cover crops or your tillage practices, etc. So we're just going to go through a couple of case studies that I've seen across the state that I thought were really interesting, and I thought were informative in how these tests reflected the different management practices. As I said, Matt already explained this. I'm going to be focusing on the soil health analyses. And this is my first case study. This is up on a farm up north of Moberly near Clifton Hills area. Southern field, it's been in a corn soybean rotation, and it was tilled quite often until about 2010. And to my knowledge, there haven't been any cover crops grown on this field. It's been farmed, described by the farmer, it was farmed pretty hard. And you can see that they implemented terraces and stuff to help with the erosion. This field and these on the right-hand side are the associated soil health tests. The field to the north of it, which is right next to it, it's under very similar soils. And the last 10 years has been managed quite similarly. However, prior to 2010, this was a grazed pasture for, I think the farmer said, for as long as I can remember. So at least 20 years. And since 2010, it was then converted into the corn soybean rotation and it's no-till. So this soil has not been tilled for decades. And what's interesting is if you look at the soil health test for this north field, you can see they immediately respond in every single aspect. So I think what this suggests, and these tests are really sensitive to if you have a perennial system and the benefits of the perennial system, that active carbon pool that Matt just discussed, a perennial system like a grazed pasture really can generate and activate that active carbon pool and expand it. And you can see that within the tests. this is like this was the easiest case study across all of them. It's really nice and easy to see the benefits of this perennial graze system and how long it has extended. Even it's 10 years afterwards, you can still see the difference compared to that field south of it. So I think it's a really good case study of how this perennial system has helped this north field. And we actually have a video where we dive in depth a little bit more. That's what we'll be showing during the break. Moving to our next case study, this is near Arrow Rock. And these are two fields that a farmer had. And this is where we get a little bit more into the nuances of these different tests. The field on the top was, they're both in a corn soybean rotation. There's lots of residue left on both of these fields. But prior to 2016, this field on the north was field cultivated prior to planting every year. Whereas the field on the south was simply, has been a no-till for decades. And these are the corresponding tests. Tests on the top compared to the tests on the bottom, you don't see any immediate huge differences like you did on that last case study. But I'd like to dive in. You can see there seems to be a benefit to the enzyme activity on the no-till system. The total protein are fairly similar. And organic matter across the field and our stamp sites were fairly similar as well. Where you see the differences in this respiration and this active carbon. Surprisingly, where this field was tilled until about 4 or 5 years ago, the respiration is higher, noticeably higher, whereas the active carbons are quite similar. So what does this mean? You hear a lot about soil health, you build up your soil health, all your soil health tests are going to go up. But in this suggested practice, no-till, it's supposed to build up your soil health. It seems like the respiration rates aren't as high. So it's kind of counterintuitive. But this kind of goes into what Matt was talking about before, where it's what is going on in your farm system, what is happening within the soil, within the microbial community. And if you look at the active carbon on the x-axis, on the y-axis, excuse me, on this area, they're quite similar. And the more active carbon or epoxy, as it's also called, the more you have that, the more storage you're providing. And then with respiration, the greater the respiration rate, you're getting nutrient release because that carbon that's in the system is being broken down and being released because the microbial activity is active. And you want an active microbial activity. And like Matt said, you want the sweet spot where you're balancing both your stabilization and storage with your nutrient release. So what's happening in this system is the field that has been a no-till is more along the stabilization side of things, whereas the field that was cultivated until recently is more along this nutrient release. So I think it helps you understand, you're getting more nutrient release, but your organic matter might not actually be stabilizing. You're not increasing your organic matter. So some of the situations where people say they've been doing no-till for a long time, and they haven't seen an increase in their total organic matter, perhaps this is the reason why. They're more on this side where they're mineralizing a lot of carbon or a lot of nutrients in their carbon pool, and they're not stabilizing as much, so they're not able to build up. Or perhaps their system with the current management practice that they have is settled more in this mineralization side of things versus the stabilization side, so their organic matter isn't actually growing. So that's one way you can interpret these tests, and it can help inform you. Hopefully these reports can help you understand your fields in a better and a new way. The next case study is similar. It's in the clay pan region outside of Mexico. And this, we're comparing some no-till plus cover crop fields to a field that uses a light tillage right before planting. And this is a little bit messy, but I'd like to focus on Where the no-till and the cover crop farmer is farming, they have quite similar soils. And clay pans have, they can be quite difficult to work with, but his organic matter and his active carbon are higher than where the light tillage is occurring. And then moving and looking at the respiration, perhaps what has happened is this farmer has decided to incorporate no-till and cover crops. And what is happening is, as he has done that for the last few decades, he has moved his system from perhaps a greater mineralization to where he's stabilizing and he's able to increase his organic matter. And that's why his organic matter is at a greater, is at the higher end, versus the farmer on the right-hand side was at the middle, and relatively to his geographic area was lower than this farmer that was running no-till and cover crops. So I think what this does is it helps us understand the different recommended management practices, how they're affecting the soil in your fields. So I would recommend looking at several of your fields if you were working with us and comparing the fields and hopefully and trying to discern why you're seeing some of the different trends that you're seeing. The last case study is outside of Marshall and Higginsville. So on some very productive soils, some really nice lush soils. And these fields are, these were manure applied. They received quite a bit of manure, and they also were cover cropped and no-tilled. And this is all within the same farm. And then on, well, sorry, excuse me, the farm on the top, and the analyses we're seeing on the right-hand side, this is just a no-till field that does not receive manure, but it is no-tilled. And to my knowledge, it doesn't receive cover crops. And as you see on the field on the south, as I go to the next slide, this is a manured field with cover crops. And you can see that the manure and the cover crops have really expanded and increased the active carbon and the respiration within the system, also the organic matter. And so these tests we're used to, I think, as agronomists and things, looking at, you know, your pH or your potassium analysis or your phosphorus, and you just look for your critical value and you're like, how far above or below am I above this? And that is, you know, a really easy turnkey mechanism to make a decision. Unfortunately, we're not at that point with these tests. Imagine yourself back in the 50s when you first heard about the Bray 1 test. You're like, you know, what is this? How do we use it? That's kind of where we're at with these soil health tests. These soil health tests have really been geared to help us understand the system and what's happening. Like what's my trajectory on these different tests, and what is going on with this field compared to other fields. And you guys know your fields better than we do. So you guys, I think with these reports and with the understanding of what these tests are, I think you could really gain a lot. Well, you probably understand more than we do because you just know your fields in a way that we, Matt and I, we don't get the cab time and the tractor driving across these fields and seeing these things. So if you have questions, feel free to reach out to us about your different reports. We're going to have a 10-minute break right now. We're going to play a video that we made. This video, Newell encouraged us to submit it for a competition within the basically the scientific community we're a part of. And we actually won a prize for it. So that's kind of cool. And anyways, we thought this would be a good breaking point. If you want to watch the video, hopefully the video is not too choppy. It depends on your internet connection.
Jeff, did you want to go ahead and field any questions first before the video and then break? Okay.
Yep, I was going to say we'll field questions, I think, for about 5 minutes and then start the video. Kind of when the questions die out, I think. So.
Yeah, so there's a couple questions in the chat. So Ed asked, how do you calculate yield? So I believe I mentioned stuff about yield in the next presentation. I don't know if Jeff, you actually talk about how you calculate yield, but that question should be answered in my next presentation, I believe.
Yeah, the short answer to that is we collected about 50 linear feet of row from each of the treatments, and we hand harvested it. So that's why there were piles of corn when you went through and you harvested. And we weighed them in a bag, and then we just basically converted that pounds per 40 square feet or 50 linear row feet to pounds per acre. So that's how we calculated yield. Was we harvested about, I think it was about I don't know, about 50 pounds of corn from each treatment. So that's how we calculated yield. Then we also, on top of that, we also calculated the moisture of each treatment as well.
And then Brent asks, how were the stamps positioned in the fields? So to answer that, there was a lot of stuff that went into it, especially this year. So last year we didn't really pay attention to access too much. So we found walking out to the stamps that were 400 meters out in the field during harvest was rather tiring. So position of, where our access was to get into the field came into that. And then we also looked at fertility maps if the farmers had them, and tried to position our stamps in places that had either low fertility or a spot with high fertility. And then we also tried to place them in different landscapes as well, so we could capture all these differences within a field. So I don't know if you have anything else to add to that, Jeff.
Yeah, and then at a larger scale, we chose the fields based on historic practices. So we tried to get a spectrum of soil fertility and then, management practices. So a lot of times we were on one with a lot of the farmers, we were on like their better ground. And then also a lot of the times on some of their rented ground, where the rented ground a lot of the time doesn't get all the inputs or is farmed a little bit differently than the ground they actually own. So most farmers will put cover crops on, the farm that they own. And then sometimes it's just more difficult putting on rented property just because you're working with the landowner and stuff. So we tried to capture a spectrum of soil fertility and soil health.
So hopefully that answers your question. Dr. Scarf asks, I saw you using yield ratio in your analyses. Are you also using yield difference? Jeff, you're the yield guy. I'll let you.
We haven't looked at yield difference. I think that's the best way to answer that. We do use a cutoff because this isn't replicated, Dr. Scarf. We use a cutoff of saying the yield had to be this different from the control for it to count as a response. So, you get a little bit of a jump around your actual average, So to say that there was a response at a treatment, it had to have a certain difference than the control, but I don't think we're using yield difference in the idea that you're talking about for our analysis. We're mostly focusing on, for the analysis we're going to talk about today, we're just going to be talking about, did it respond or did it not? We're not going to be talking about the actual percent response. We plan on doing that at a later date, but I'll talk about that in about 20 minutes, 30 minutes in depth.
And then John Jones, how variable were some soil health associated tests compared to basic tests? So like your Bray 1 within a stamp.
So we only took one sample per stamp, if that makes sense. So we go and pull like 8 cores. So we only have one, I guess, number for each stamp. But I guess your question is, how variable is your testing within the soil health realm? Like how variable are these tests if you sample in like March or in June, I think is maybe what you're saying. What's the temporal variability and what's the spatial variability associated with these tests? Our data set isn't we can't answer that, but from the literature that I've read, they're similar in terms of temporal and then also spatial variability.
Well, if nobody else has any questions, I think we're good to move on to the break in the video.
Okay. Life on Earth depends upon productive soils. In recent decades, humanity has begun to realize the importance of highly functioning soil on human health. But how does one meaningfully quantify soil function? From this question, the concepts of soil health emerged. Our research focus is connecting soil health concepts with crop production. Some have offered innovative ways to measure soil health that are, perhaps, not very quantitative.
The goals of this research are to quantitatively measure the soil health, and by doing that, we want to be able to show how the productivity differs between different fields.
We chose 2 adjacent fields to demonstrate the objectives of our research. Today, both fields are in no-till corn-soybean rotations. But prior to 2011,
the North Field has been in pasture and/or hay for its history up until the last 10 years when we've been row cropping it in a no-till system, never been tilled. The South Field was in a conventional intensive farming system for all of my life as far as I'm aware of.
we wanted to delve in and see if we could understand more about the soil and how the function of that soil has been influenced by the prior historical management of these fields. In the spring of 2019, soil health samples were collected at 4 sample locations within each field prior to planting. We then processed these samples and analyzed these samples following standard soil health and soil biology procedures. The soil health tests that we measured were
Active carbon, soil respiration, total protein, and then we did a suite of enzymes.
historically, carbon measurements have just looked at how much carbon is in the soil. What these tests do is they help us look at the different fractions or different pools of carbon within the soil. We wanted to measure the active carbon pool because it quickly responds to soil disturbances and management changes. And the suite of enzymes help us understand how nutrients are cycling between these carbon pools. These quantifiable measurements give a clear picture of the improved soil health and function on the northern field.
The soil health test showed that there was a 2 to 3 times increase
In the active carbon, soil respiration, soil protein, and enzyme measurements as compared to the southern field. Management practices for the 2 fields have been similar for the past 9 years. Therefore, the differences in these biological tests are attributed to residual effects from the different crop rotation and tillage practices. The southern field, you can tell that the history of erosion has disrupted the carbon cycle and has not allowed the storage and the buildup of that carbon and those different pools within that system. These effects are still present after 11 years of no-till. In contrast, the improved biological effects of a perennial hay system remain after 9 years of no-till corn-soybean rotation. This demonstrates the ability of no-till to maintain soil biological activity nearly a decade after a perennial production system. However, the reduced biological activity in the southern field indicates that no-till alone is not sufficient to restore the degraded soil. Additional restorative practices, such as cover crops or a perennial rotation crop, might be required to restore the soil to its previous function.
So right now you can go tell a farmer to increase his soil health, but what does that really mean to him? So we want to show how increasing your soil health may increase the productivity of the soil, which to a farmer, that's going to be more beneficial than anything.
Comparing the last 10 years of corn and soybean yield maps demonstrates the dramatic difference in productivity between these 2 fields. As the yield maps exhibit, the lowest yield of the northern field is equal to or greater than the highest yield in the southern field. This demonstrates how prior management practices have influenced today's yield potential and how this should help inform farmers in their future management decisions. These soil health tests help us understand how the soil functions, and adoption of these tests can help farmers make sustainable management decisions that can last for generations to come. I'd just like to say thanks to Ryan Britt for working with us on that case well, basically on those 2 fields. He's a farmer. I think he's in attendance today. He was really helpful in helping and first providing the fields and then also providing the historic yield maps, and just he was a great farmer to work with, and really appreciate him letting us use his data and use his fields to make that video. And yes, I will post that link in the chat right now.
So we gonna take a 5-minute break then at this point? Jeff?
Yeah, I think we'll get started about in about 5 minutes. So just before 5 after. If anyone wants to take a break, go to the bathroom. I'm gonna drink some water. So yeah.
Hey, Brent.
Yeah, good morning. Sorry, I didn't realize my mic was on.
No, I thought you were getting on to say hi and visit for a second
Well,
here.
hello. That's
So-
yeah. There's a lot of really familiar names on this call. It's neat to
Yeah.
see.
Yeah, and you're
Going
gonna
from
really
that
enjoy the
perspective.
second half of this, just by virtue of some of the data analysis steps that
It's
Jeff
great.
and Matt have done. So I think you're gonna like how this is shaping out. So not to make this a teaser for the rest of the group, but our conversation will be that, I guess.
Yeah, it's really exciting to see the results here. And one thing I think is I wonder if we'll get to see a dot where all the positions or all the points are at. It's obviously a lot of work to have actually accomplished this experiment. Really impressive, actually.
Yeah, I think you're gonna see that within the next 5
Pretty
minutes.
soon here. Yeah.
Yeah. And actually, the map that you're gonna see, and I don't think we touch on this too much, but we've worked a little bit up in northern Iowa the first year, and in addition to that, been working with Jason Clark at South Dakota. And so he has a kind of a sister project. Not sure exactly how well the numbers between the two different states are gonna come together, but he's got a procedurally, we're doing the same kind of thing, and he's doing that with a graduate student up there in South Dakota. So
Well, it's 5 after, so I guess I'll go ahead and get started on the second presentation here, my second presentation here. As long as nobody has any objections. Okay, so for my second presentation, I'm gonna kind of go over the project overview. So what do we, or how do we do the stuff that we did? And then the second part of this presentation will be more of the tissue results. So let's jump right into it. So how do we do this research? So I split it up into 5 basic steps here. So first was soil sampling, then we added fertilizer treatments, and then we have our soil analysis, and then we have evaluating the plant response to those fertilizer treatments by the tissue testing, and then we evaluated a yield response to those added fertilizer treatments. So what's really neat is just looking at this picture here, this is I believe this is before we actually did the tissue sampling, but you can see this dark green spot here. So every stamp in this one field here has this dark green spot, and they're all sulfur responses. So I'll talk about that a little bit more here in a little bit, but just a neat picture here to be able to see how some plants can respond to the added fertilizer. So here you go, Brent. Here's a picture of all of the fields that we were in Missouri throughout the 3 years of this project. So 2018 was more of a trial year to make sure that everything was going to work out. That was before Jeff and I were actually here. So they only did 5 fields that year, but it was, like I said, a trial year. So in the next couple of years, we did 47 fields and 45 fields. And then with the amount of stamps, so we placed, multiple stamps in each field. So in 2019 in Missouri, we had 195 stamps. And then in 2020, we upped it to 238 stamps. So overall for the project in just Missouri, we had almost 100 fields and almost 450 total stamps. So on top of that, we had, I believe, 10 or 12 stamps in Iowa last year. And up in South Dakota, I'm not sure the number of stamps that they have, but I want to say it's between 75 and 100 stamps for the 2 years combined. But so that data is a little different than the Missouri data. I'm sure you all can imagine how different the soils are. So what's pretty neat is the distribution that we have here across the state. So I have included this map of the different MLRA soil types across Missouri. So as you can see, we're in a very we're very distributed across the state in terms of how many different soil types that we're actually in. So some of these will be able to compare within the soil type later on. We're not there yet, but yeah, so it's just neat to see the different distribution across Missouri that we were able to obtain in these 2 years of research, 3 years of research. So now I'm just going to show some pictures of the soil sampling. So there's me putting out the stamp outline. This is a picture of me and Newell. And as you can see in the middle of this picture, we have a GPS. So with every point, we had to go out when we soil sampled and took a GPS point. That way we were able to come back later in the year. So that was pretty neat getting to use that. And you have your different soil probes. And then we took deep cores when we soil sampled. So this is a picture of the machine. I'm sure most of you have seen this, you farmers. What this does is it goes down to about, I think 3 foot. So we can take a deep core and we can analyze that, which I'll talk about here in a little bit. And then after that, after we took these samples, we applied fertilizer. So it was very important that we kept the fertilizer separate from these soil samples because we didn't want to contaminate anything for those fertility samples. So I came in after, whoever had taken the soil samples, and I spread these fertilizer, and I kind of talked about the different treatments that we had. But the reason, so it says no fertilizer for treatment 1, but we actually applied some nitrogen because in our treatment 4, in the sulfur, there was ammonium sulfate. Ammonium has nitrogen in it. So we wanted to keep everything equal. So we applied the same amount of nitrogen to each stamp or to each treatment that was, coming in that treatment 4, just inherently. So you've already seen this picture, but it was a good time. I think I had a good time. I'm sure Jeff did too, doing the soil sampling. So just got this short video here of the soil sampling, and oh, never mind. Guess it's might not work. Me spreading fertilizer in the background. So obviously he's not taking soil samples out of our stamp. This is just a video demonstrating, what we did. But I'm spreading fertilizer here after he had already taken soil samples out of that stamp. So you see there's 2 buckets here. Why do we have those 2 buckets? So now we're going to move on to the soil analysis, and I'll explain why we had those 2 separate buckets. So first off, we collected fertility samples. So we had 0 to 6 inch fertility samples, and we sent those off to Ward Labs and just got the fertility ran on those, so we could obtain all those results or all those fertility readings that were in all those reports. And then we had the soil health samples. So with the soil health samples, we split those up, which is why there was the 2 buckets. We have the 0 to 2 inches and then 2 to 6 inch samples. So what this does is it shows the difference in that in these soil health tests. There's some stratification that can occur because of no-till. So we decided to split these up to be able to show that stratification that can occur. And also we took those soil profiles, so those deep cores. And what we're doing with those deep cores, what we're able to do is show these different horizons and how deep each horizon is. And we also ran soil fertility on the top 3 horizons and the texture. So we're able to obtain some information about those horizons that are a little deeper than just those 6 inches. We haven't gotten to analyzing any of this yet, but eventually we will. So I wanted to mention that the soil health samples were ran in the USDA lab. They're at the Ag Engineering Building on campus, and me and Jeff have ran most of those soil health samples, but it definitely would not be possible without Jill and everyone else helping us in the lab. So we wanted to make sure we mentioned Jill and Kristen and everyone in the lab. So after we did after that step came tissue testing. So we took tissue tests when the corn was at the V6 growth stage, and we took the whole plant. So why do we do the V6 stage? So the V6 growth stage is right before a lot of the reproductive parts are being established in that V7, V8 stage, you know. So it's a very important growth stage, and it's been shown in research that sampling at this stage can be beneficial to predict your yield, which you'll find out later. Maybe not always be the case, but it's an important growth stage, which is why we chose to tissue sample there. So what we did was, in each treatment, we took 8 plants. So in each treatment, in treatment 1, treatment 2, treatment 3, treatment 4, we took 8 plants, and we took those back and we weighed them and then dried them and then weighed them again to get the mass. And then we, so after that, we ground them up and send them off to be analyzed for the tissue concentration of each nutrient. So that way we could run these analyses. And then after tissue, there was the harvesting of the corn. So this is going to get into Ed's question as well. So after that, after the corn reached black layer, we were able to go out and harvest. We like to wait as long as possible to make sure to get the corn drier so it's easier to harvest. But sometimes that wasn't always possible. So what we did was we went out and each treatment, we took 48 linear feet of corn, and we weighed that. So we weighed that in these white bags. And after we weighed it, we dumped it, and we took eight ears sample. So we took 8 ears out of each treatment. And what these 8 ears, well, there's only 4 here, but imagine there's 8. So we took 8, and what we did with those was we weighed them before we dried them, and then we dried them and weighed them again to get the moisture content. So we have the weight of these 48 linear feet, and we have the moisture content. And using those values, we were able to convert to a yield, as Jeff kind of talked about. So that's what we did for the harvesting part. So now I'm going to kind of get into some results. So this is why we're all here. So first off, I'm going to start with some of these response graphs. So if you attended last year, these look kind of familiar. So this is including all of the data from 2019 and 2020 in it, and I believe 2018 is in it as well. So these response ratios, which is what's shown on this graph, are calculated the same way that I explained earlier. So for the tissue, we took, for the potassium tissue, we took the potassium concentration out of that potassium treatment and divided it by the potassium concentration out of the control. So that's going to give us a response ratio over here on this side. And then down here, you just have your soil test potassium in parts per million. If you're familiar with this in pounds per acre, you just basically take these and multiply them by 2. So then what I told the program to do was to put a best fit linear plateau graph in. So basically this red line is showing the best fit of all this data. So it comes to this point here, which is interesting because if you add in what the critical value is, it's pretty close. So what this black line here is doing is showing the critical value that Missouri has established for potassium. Now, if you know anything about it, about the potassium critical value, you know that it depends on the CEC. So this critical value can be moved around a little bit based off the CEC of your soil. So you could be below it or above it for your critical value for your specific field. So I just did like an average of our CECs to put this line in here. So I wanted to show was how close this actually is to that critical value. So basically what this is saying is if you're beneath that critical value, you're going to get a tissue response to added potassium most of the time. Or it's more likely that you'll get a tissue response. Now there's still some points down here that, you know, don't respond, but hopefully the soil health test can try to explain those points as to why they didn't respond to the added potassium. So it's just really interesting how close it is to the critical value that's already established in Missouri. So the next, if we move on to phosphorus, a little more scattered here than the potassium, but also that the critical value for phosphorus is 22 or 44 pounds per acre, 45 pounds per acre. So that's where that black dashed line is at 22 parts per million. And that you see again, it's right there on the dot. So what this is telling us is that what we're doing is actually showing the correct stuff, and that the established Missouri
Matt, we lost you here on the. Hey, Jeff, can you hear me?
Yeah, I can hear you. I think his audio went out.
Yeah, for some reason you're on.
Okay, I can hear you now. Can you hear me now?
Yeah, we got you back.
Okay. I don't know why that happened. That was weird. So did you guys miss anything there? Like, was I moving through the slides without you guys hear, or did you guys hear me the whole time?
Just about 10 seconds worth is all, so.
Okay. So it's just interesting that these both lined up near that Missouri critical value. So I just wanted to show that what we're doing is a real response, and it's pretty interesting to see that for potassium and phosphorus. Now sulfur is a different story. So I think everyone probably knows that the sulfur sulfate test is pretty variable, I would say. So there's not really a trend in the data here. It's pretty much a shotgun pattern. I kind of put in this critical value that Missouri has, but there's not really a trend whether or not you get a sulfur response to added sulfur. So I think with that, I'm gonna move on to Curtis. Has a couple slides here, if he's ready.
Should I just request control, or are you gonna click through them?
I can click through them if you want.
Okay. So Jeff and Matt asked me to explain a bit of their methods. In the previous graph, Matt showed there was just this huge scatter, and sometimes it responded even if it was above that critical line. And so they will be talking about a few of their results in terms of explaining some of that scatter. And they're hoping that I would walk through and describe one of the decision trees that they're using. But before I do that, I want to backtrack to the objective. Again, the objective of the project was to look at soil health and its impact on improving how we manage soil fertility. As you go to the next. So there's two ways we could have gone about doing that in discussions with Newell earlier on. We could have done the classical plot experimental design where we have a lot of replicates in a single area. And the benefit of doing it that way is that we would have added confidence. If we did see a response, we'd be confident in that response. However, a lot of our resources would be towards building these replicates. And we wanted to expand and get as many different environments and as many different measurements as possible. So we went with this other approach, and I'm terming it as machine learning. And essentially, as Matt described, you only have one little stamp in an area. You don't have a replicate, but there's lots of stamps throughout a field or across many environments. And again, this lets us take a lot of measurements. You can go to the next slide. And essentially what we want to do is get this big pile of data. And the hope is that we can use some machine learning techniques to kind of filter through this data and figure out what soil health or soil physical attributes might help explain where it was responsive or where it wasn't responsive, even though the critical values say it shouldn't be responsive. And so just to backtrack a little bit, machine learning is used quite a bit. And one of the definitions of machine learning is a computer algorithm that improve automatically through experience and the use of data. So as we grow our data set, the possibility of these machine learning algorithms to help us improves a lot. And just to reiterate, the goal that we're using these machine learning algorithms are to filter through these data and identify the important variables. Now you can go to the next slide. So one algorithm that Matt and Jeff have used is this decision tree. I'm going to go through an example and how to interpret it. So here on the right-hand side, we have red squares and green circles indicating yes, it responded, or no, it didn't respond. And how the decision tree works, it tries to partition the data into equal groups. So for instance, you'll sort through and find a variable and say, hey, this variable helped divide the data into two. So in this example, tillage might have helped divide it into no-tilled or tilled, divided into two separate groups. But we can further partition the data into additional groups, including other information. So this decision tree or tree will grow. And for your information, the first split tends to be the more important variable. And then as the decision tree grows, those subsequent splits are important variables, but at a lesser extent. So you can see here how to read the tree. If it was tilled, then you would base your prediction of whether it would respond or not respond on a soil test value. In this case, I just put potassium as an example. So whether it's high or low, it would indicate where that partition occurs. Then on the flip side, you can further divide the data up based on the no-till, and this would be on some biological activity, whether it was high or low, you could then get the split. So Matt and Jeff are going to be showing a lot of these decision trees, and so as you read them, you just start from the top and go down, and pretty logical way of reading through them. But you can tell that with no-till, whatever variables under that is linked to no-till, whereas with till, whatever variable under that is linked to till. And I think, Matt, you're going to explain, even though we didn't have replicates, Jeff and Matt took a pretty conservative approach to identifying when there was a response and when there wasn't a response. And I'll let Matt describe that.
Okay, so thanks, Curtis, for explaining decision trees. So for my analysis, based off the tissue response versus no response, I did a 5% response. So if it was under 5% response ratio, it's classified as a no response. But if it was over 5%, then we classified it as a response. So we did that for this decision tree. So this is a decision tree for the potassium. So basically what this does is we put all our soil health variables in here and all of our pH or our fertility variables in here. And this is basically telling us how we can get a response or a no response. So what's interesting is before I jump into this tree and trying to explain it, if we just took the soil test of potassium and predicted a response or a no response, it was 65% accurate in predicting that response. But this decision tree, using this, the accuracy went up to 75%. So I'll talk about that here in a second. But so basically, if we go through this decision tree, what it's telling us is at a soil test potassium of 119, if you're lower than that, so if you're under basically the critical value, then you'll get a response. And 46% of the data was in here. So, but if you're above that, there's a couple different things that go into it to decide whether or not you'll get a response or no response. So if you're above that critical value of 120, then it moves on to your soil respiration value for your field. So this soil respiration value of 234 is actually extremely high. So our average soil respiration was around 140 of this variable, or this, sorry, our average soil respiration was around 140. So this 234 was really, really high. So if you had a high soil test K and a really high soil respiration, then you're gonna get no response to added fertilizer. But if you're under this really, really high value, then it moves on to one of the enzymes that we measured. So with this phosphatase value, 185 was actually the average. So this is, right in the middle of our phosphatase values. So if you're higher than this 120 soil test K and you're lower than this soil respiration, then it goes to your phosphatase value. And you can read, so if it's less than the average phosphatase value, you get a response. If it's greater than the average phosphatase value, you don't get a response. So this is a more complex way of looking at it than just looking at your fertility value and just saying, well, if I'm below that, I'll get a response, or if I'm above it, I won't get a response. This kind of takes in our soil health value or soil health measurements and is able to better predict whether or not you'll get a tissue response. Remember, this is just tissue. This doesn't have anything to do with yield yet. So the take home from this decision tree is that a combination of soil fertility and soil health tests may be more accurate than just using the fertility when you're making your fertility decisions in the future for tissue responses. And then the last thing that I have is tissue response versus a yield response. So basically, if there was a tissue response, was there a yield response? Because, if you get a tissue response but no yield response, then as a farmer, what does that really do for you in the end? So these are just I'm just going to run through the potassium. Hold on. I need to mention that this is preliminary. This isn't a final decision tree. We still have some tests to run. And I only talked about the potassium one because it was one that made the most sense at this point in time. But we are going to do the same thing for the phosphorus and sulfur when we get everything done. So just wanted to make that note. And then for this part of it, I'm only showing the potassium again, but we will do the same thing for the phosphorus and the sulfur. So jumping into this, so 23% of our stamps had no response to tissue at the tissue level or the yield level. And then so 44% of our stamps did respond to tissue, but did not respond to yield. So what this is saying is at that B6 level, the concentration is higher, but then when you get to the yield at the end of the year, there wasn't a significant difference. And then we'll throw on yield response here. So at 11% of our sites, we did get a response, but we did get a response, which is pretty interesting. And then 22% of the time, we saw both a tissue response and a yield response. So what this is telling us is that 45% of the time, your tissue response or no response will correlate with the yield response or no response. So hopefully I can incorporate some soil health stuff in here to try to explain why this is. But at this point in time, all I can say is that the tissue and the tissue response or tissue test doesn't always mean that your yield test will be the same. So with that, if you have questions, you can write them in the chat, and I think we'll field questions at the very end. So I think with that, I'm good to go on to Jeff's presentation over yield results.
Thanks, Matt. Yeah, so I'm going to be covering the yield portion, similar approach to what Matt took and what Curtis did a great job explaining. And yeah, hopefully we can, after this, we can have a good discussion on what we're observing. And I'd like to first bring out, I'm not sharing my screen, I don't think. There we go. I'd like to first bring up what Curtis said is we did not have replication. So we didn't have a bunch of measurements in the same area. We just had one observation in each area. And the way that we got about understanding how much error was associated with where we sampled, I'm going to talk about that. Typically, the way that we get across this by doing, maybe you guys have done this, is, strip plot trials. You get lots of observations and you have a comparison right next to each other and you do it a couple of times in the field. We did not do that. So the way that we were able to know or say that there was a true response in each location, each of the locations we were in, was we asked several of you, we got about 20 fields of yield map. And what we did is we went to our stamp locations and we extracted out about 50 by 50 meters. And we just looked at the variation in yield in this area. So what is the inherent variability? And we looked across all 20 fields. And what we came up with was, if we had a 13 bushel per acre increase from one of our treatments, we were confident that that was a response. So from everything that I'm talking about, if I said that there was a response, it's because there was a 13 bushel per acre or more response. And we basically just divided that into, yes, there was a response to our fertilizer application, or no, there wasn't. So that's what these results are based on. So 13 bushel per acre, some of you are probably thinking that's pretty high. And yeah, it is. So this is a fairly conservative approach to this analysis. Ranges, if you got about 150 bushel per acre to 200 bushel per acre, around 6 to 9% of your total yield. So I just wanted to get that out there so that when we're talking about these results, you can keep that 13 bushels per acre in the back of your head. And I'm going to be talking about which variables were important from our analysis. And I'm going to be talking about them within our different treatments. So this on the top, the 1 through 5, number 1 was the most important variable that we used in predicting our results, our response to a potassium fertilizer application. So the variable in the number 1 slot was the most important. The variable in the 5th spot was still important, but not as important as the variable in the first cell in the number 1 ranking. And the variables that we chose were, or not variables, the soil samples that we chose were ones like the standard soil chemical tests or fertility tests, then also these suggested soil health tests that should be included in a soil fertility decision or fertilizer application. And I'd like to highlight within our potassium and phosphorus, the 2 most important variables for both of those analyses was the soil test potassium ammonium acetate analysis and the Bray one, which I think is a good thing, because we know that these tests work. There's been a lot of research put into them, and it's been established that these tests are a good indicator of how much is available to the plant during the course of the season. And so I found that comforting because it meant that, you know, our data set, we are observing real things. And I think what we want to look at is the nuances of when the Bray-1 test or when the ammonium acetate test are not very good indicators of when you're going to see a fertility response to your, not a fertility response, a yield response when you apply fertilizer. The other important variables were pH and then also total organic matter. So, you know, standard soil fertility, I highly recommend still doing it. I'm not saying toss it out. We just want to see if you can refine it with some of these soil health analyses and variables. And those were included. And if you remember, they measure a specific pool within the organic matter in the soil. And our active carbon was in each of the different treatments that we applied, soil respiration, and then also total protein, and then also our enzyme activity. So a lot of the analyses that are being suggested to be included in fertility decisions were in some of the most important variables. But at what value? This just tells us they're important in helping us understand when or where we're going to see a response. And that's getting back into what Matt was talking about with those decision trees. You can think of a critical value of 45 pounds per acre for your Bray-1 test kind of as a decision tree. It's like, are you below it or are you above it? These basically, the way that I think about these is, you know, I have my Bray-1 test. What else do I need? I wanted to talk about potassium first. And as I said before, that potassium, the ammonium acetate test was the most important break, followed by soil respiration. And I'd like to say that if we just used our ammonium acetate test like we've been doing for a long time, our accuracy was about 48% if we just used that critical value of our ammonium acetate and our CEC. If we did our soil test K plus our soil respiration, it increased our predictability of when we were going to see a yield. We went up to 68%. So our accuracy improved significantly by including this soil respiration test. When we did it for phosphorus, the Bray-1 test was the best test. So phosphorus did not improve when we were making an actual decision. You know, the other variables are important, but when it came to like making a decision and informing a decision, the Bray-1 test was sufficient. And we're going to look further into this, but as of right now, with the assumptions we've made, the Bray-1 test is sufficient in describing when you will see a phosphorus response. So I thought that was very interesting. And then with the sulfur, the tests that were the most helpful were actually the tests that sampled the active carbon pool, and the total protein, the active carbon, and soil respiration. And so the state sulfur test was about 50% accurate, which is kind of, you know, flipping a coin whether or not you should apply fertilizer. Whereas the soil health metrics bumped it up to 63% and improved it a significant amount, I think. So we demonstrated, you know, well, first I want to talk about how you might, you know, make a decision based on this. And this is what we want to go to. I'm not going to say this is, you know, take this to your fertilizer dealer or to your agronomist and say, this is what I want to do now. But this is how we hopefully will be, what we'll hopefully be able to do near the end of this project, and how it might help farmers understand when and where to apply fertilizer. So this is the nutrient concentration. I'm focusing on potassium. This is the critical value as Matt described. There's a little bit of variability based on your soil texture and your cation exchange capacity. But for simplicity's sake, this is, you know, a good demonstration. And then if we incorporate our decision tree into it, what our decision tree or our data is suggesting is that critical value might be a little bit lower than what the current state recommendation is. We can get into and debate how much, and hopefully during the end of this, we'll be able to say a little bit more confidently how much, but as of right now, it's suggesting it's a little bit lower than what the state recommendations are. And then when you include the soil respiration data, if you are below the critical value and you have a low soil respiration, so your biology is diminished or lower then you're more likely to see a yield response. So what's interesting is if you have a soil test that is less than the critical value, but you have a high biological test or soil respiration test, that can overcome the deficiency that is suggested through the soil test. So if your soil test K says you're really low, but you have high microbial activity or high soil respiration, that can overcome some of those deficiencies. And then on the other hand, if you're above that level, you can often will see see yield responses when your soil biology is lower than the average. So I don't want to get into specific numbers, as I said before, but I think a way that you could think about this is, look at those reports. If you have a really low pounds per acre on your potassium test, as demonstrated here, the sites that you're going to see more likely to not see a yield response is at those sites 5 and 1 moving forward. So that's an idea, and that's where we hope to be able to get. And really, this data set might not even be able to get there, but one day, if people continue to look at this, this is what we're suggesting. And it's specific to, right now, the potassium and the sulfur test, not necessarily the phosphorus test. And I'd like to keep in mind this 13 bushel per acre response that we're indicating. So yeah, I think it's important to understand the assumptions that we have associated with the results that we have. So we know that some of these soil health variables are important. The question is, these soil health variables or analyses were created so that we could understand what's happening with the soil based on management practices. So we wanted to look and we did the same thing, but instead of predicting yield or understanding the variation in yield response to fertilizer application, we looked at management practices. And in addition to that, we also looked at different inherent soil characteristics, such as climate, so like seasonal variability. We looked at what was going on in the soil profile, so your soil series information, and we looked at where you're located in the state, or the MLRA, so that we understood, there's a lot of variability across the state. What does this mean for informing our yield response to our fertilizer applications? And there's a lot going on in this graph, and I'm going to break it down really quick. But on the left-hand side, we have our analyses, and on the top, we have our rankings of variable importance. So I think something that's really interesting is year and your percent clay, or what you can think of as your texture. They came out to be the most important variables with understanding your soil respiration values, or your total protein values, or your active carbon, or your enzyme activity. Unfortunately, those are things that you really can't control as a farmer. And they came out as the number 1 and number 2 ranked variables. Things that also farmers can't control is the MLRA, or the location that you are at the state. So if you're in, like, for example, a river bottom field, or you're in the Mississippi, alluvial loess deposit area, Hannibal compared to, Higginsville, those soils just behave very differently. And that's what I was describing right there, those locations across the state. And then also included were the soil series information and the drainage class. I accidentally am missing a slide. About 50% of these variables that are included as important from our analyses, farmers can't control. But the ones that you can control, such as, your fertilizer source, if you're applying such as manure versus synthetic, those were very important in soil respiration, total protein, and active carbon. Cover crops was actually included in every soil health analysis. They seem to be sensitive to cover, whether or not you're growing cover crops. Oh, there's that slide. And then also rotation and tillage were included in several of these analyses. So the management practices that are promoted to increase soil health, they seem to be important, but what really seems to be more dominant in creating these baseline soil health analyses is where you're located across the state. And so one thing that I'd like to bring out, and what this means for you as a farmer, is looking at these MLRAs, or where you're located in the state, and different soil characteristics that you're going to have in the state. If you look back at this soil this decision tree for where you're going to see a potassium response. And then you look at the respiration as it broken out across MLRA. So these are different locations where you're at in the state. If that cutoff value is 120, is about 120, you can see where you're at located in the state. Achieving a respiration rate of 120 is in a clay pan region is actually quite easier to achieve than if, for example, you're in an Ozark Highland area or in the wooded slopes. So if you're like on an alpha sol versus a mollisol. So when these soil health indicators, and as we learn more about them, what we're finding is management practices do influence them, but you have to first account, I think, for where you're located and what your soil, physical properties of your soil, what's your texture, where are you located? And so this might help fine tune where we're seeing responses. And like I said, this is based on our data set that we've collected. And I anticipate with research moving forward and the big push that's really happening in soil health and soil sequestration, there's going to be a lot more information of, the variability associated with these different soil health tests based on where you're geographically located. John Jones asked what the variability of the soil health tests are compared to, your soil fertility test. The nice thing about soil fertility test is it seems to work fairly well across the state. They've developed it that way. And you can kind of overcome if you're deficient in your potassium by applying fertilizer. With these things, I think we're going to have a harder time adjusting soil respiration rates. It's going to take longer because it's just a change in management system. And it's harder to overcome some of the inherent differences within soils across the state with improving these soil health tests. Not that they can't happen, because management practices are important and can help, but you have to keep those numbers. You have to understand them within the context of where you're located. And so those are kind of the conclusions. It appears, and our data suggests that soil health tests can improve understanding soil fertility recommendations or where we're going to see responses. But like I said, the old dog is doing a good job. The stuff that we developed in the 50s and 60s is still really important. And all this, the soil health tests are doing is they're helping us understand where maybe these, chemical tests or where these soil fertility recommendations are maybe not as, maybe they break apart, or maybe they're not as accurate. And then all conservative management practices improve these soil health tests, but the baseline values seem to reflect the inherent soil characteristics based on where you're located within the state and your different soil properties there. So, with that, I think we're welcome to field any questions. Matt, if you want to go through the chat. And I think a lot of the people on this video call they're boots on the ground. They're the agronomists and they're the farmers. And so, if you have some questions or if you have some ideas of things that we might not have thought of, feel free to shout them out. We hope this will be somewhat of a discussion, and we'll be able to hopefully understand and bring to light and, hopefully understand how these soil health and soil fertility test analysis can interact.
So it looks like John Jones asked, what would you expect the decision tree to look like if both soil test K and tissue K were used to predict a response? So that's interesting. So as we saw in my presentation, the tissue K response under half the time was correct in predicting that, yield response. But if it was used in conjunction with the soil test potassium, that it might be able to explain some more there. So that would be interesting. I didn't do that for this meeting, but I definitely was planning on doing that. So yeah, it might be helpful in predicting a yield response or not.
Yeah, I think the question will be if it helps explain if that tissue will help explain where you're going to see response better than some of the other variables that are sampled, if that makes sense. So all these soil health tests and all these soil chemical tests were taken at the beginning of the season. So, maybe you can use those as a beginning reference, as I'm thinking as a farmer, as a beginning reference. And then a lot of these farmers use tissue to fine tune later. But it'll be interesting to see if it does a better job predicting where we're seeing yield response compared to these other soil samples that we've been doing.
Yeah, and tissue testing costs quite a bit too. So you need that value to be there in order for it to be a valuable thing for the farmer to do. So yeah, that would be interesting. So Curtis asks, how much do some of these soil health tests cost? You can answer that, Jeff.
You can look at the Shack, which I have their cost actually pinned on my office. A basic soil health package through the Soil Health Assessment Center here at the University of Missouri. It's $36, and it includes a couple of analyses that we didn't include in our data set, but it includes that active carbon test that you're looking at. And then if you do an expanded soil health package, it expands to include a couple of other ones, such as aggregate stability, and that's running about $80. So the soil health tests are going to be more expensive than a soil fertility. I know some of the commercial labs, such as Ward Laboratories, and I don't know if anyone here besides the Shack is doing any soil health analyses. They're going to be more expensive than the fertility ones. I think just because the throughput hasn't been developed for these tests yet, like it has. So the economics associated with running a lot of these samples hasn't been developed yet. So they are going to be more expensive. And if you're interested in using them to track where you're going with your fields, it might be interesting to sample on multiple years, just to see where your field is headed, because we saw that there's some seasonal variability in these tests.
So Noel asks, what other factors do you plan on including in the future decision tree analyses? Example, subsoil fertility. So I know, at least I think we both plan on adding subsoil fertility in there. There's I believe one other soil health analysis that needs to be included that we're not done yet. We're still running that one in the lab.
We want to include weather variables, so that we might be able to understand, which weather patterns are affecting these different tests, and where we're not seeing, yield response, because weather is a really, really important factor in yield production. So we want to be able to include some of those weather variables, such as how much rainfall, the distribution of the rainfall, things like that. We also are, we look, we included texture. So soil physical properties, we've included texture, and then drainage class. Sorry, we've already done that. So I guess that's already a part of our analysis.
And John Jones asks, thoughts on using a system like this to calibrate for a rate decision? What do you think, Jeff?
I think you'll need a different data set to answer that question, because we just had a fixed rate of 100 pounds of P2O5 or 100 pounds of K2O. Our data set is geared towards answering where are we seeing a response and why. I think to be able to do a rate response or a rate application following this, you'll have to look at these things, kind of do a rate study to some effect. And maybe the approach to doing that is to going into the different environments and like the clay pan versus like the Lost Hills near Higginsville, and then doing some rate studies in different, management system and soil health conditions, so that then you would be able to do a rates recommendation. But as of right now, I don't think our data set can answer that question because we're limited by the fixed fertilizer application. So we're more geared towards answering, do these soil health factors, are they helpful in understanding soil fertility?
Does anyone else have any questions? Thanks, John, for the questions. They're good questions.
Yeah.
And everyone else.
A point that I will add here is that, I think the two of these two have done a wonderful job. As they've mentioned, they still got their feet in the laboratory, finishing up some of the results, and to be able to kind of push it to some level of first analysis needs to be commended, but it's also just kind of a teaser showing that these relationships are for real, they're empirical. We can show that biology does matter when it comes to fertility decisions. It's secondary, but so as they complete their lab analysis and complete the numerical analysis over the next year, we expect some really interesting, more well-developed messages that probably the group would love to have right now. But it is what it is in terms of what we can do. We're running these two are running as hard as they can to get this done. But I would anticipate that later this year, as a part of crop management conferences, as a part of the different grower meetings that might be throughout the state and elsewhere, that we'll have a refinement of this message and more specifics that would be offered in terms of how it might be more universally utilized. So, but we put it as a goal to ourselves, so we were going to cook it as far as we could at this point, and allow them to kind of see that this is real and it's has some value. And so hopefully, at least from my standpoint, I think they've done a great job.
I don't know. I'm interested to hear from if there's any agronomists or farmers that, if you have any questions, or I don't know, maybe some kickback, or, not kickback necessarily, but, a good healthy debate on what we're observing and why we might see different things. Or if you have questions about your report and your specific fields, we'd be fine to field those right now. If you're busy and you'd like to talk to us at a later date, you've got Matt and I's phone numbers. We are very willing to meet with you in person or over phone or Zoom. Well, I don't know, with COVID restrictions, I don't know if we can meet in person right now, but over Zoom would be great.
Well, with that, I think that that pretty much wraps us up. Unless anyone else has anything to say, we would like to, of course, thank everyone for joining today and listening to us ramble about these results and everything going on. So we do have one more question. Linus asked, from what you have learned, what would you look at in the future in terms of what we look at in terms of different variables? That's a good question. What you look at? So one of the things that we are planning on looking at, pending funding, is the grain response.
Grain
By
nutrient.
grain response, I mean the grain nutrient content. Sorry. So we still haven't sent our grain off yet to be analyzed for the nutrient content of it, which could be pretty interesting in terms of, how much of each nutrient is in the grain. So pending funding, we're still waiting on that.
Yeah, I think within the realm of the sulfur, we hear a lot about sulfur and how we need to apply more sulfur. And I think some people are seeing it benefits more than others. I think I would like to look at sulfur and also how that might interact and where you're seeing a sulfur response with these soil health indicators based on your nitrogen applications. And that might help us understand the sulfur portion of these a little bit more. I think that's one thing that I'd like to look at, Linus. And then going back to John Jones' question, I think for this to be helpful, we need to get to where we can fine tune fertilizer rates. So I think we need to get the next step after this would be doing a rates thing. And while I'm talking about it, the other thing is I think we need a calibrated per region soil health baseline so that farmers know it. Because right now, your soil health numbers are, they're just for your farm. You don't have at the end of the year, you get like a county yield average for the yield. So you kind of know where you're at in relation to the yield. It'd be really nice if we had something like that for these different soil health indicators. And we have, it'd just be really helpful, I think, for farmers to be able to inform their decisions if they're going to use these soil health indicators in a better way. I think until we really understand values and thresholds in a better and more comprehensive way, these soil health tests are going to be, in terms of implementing them on farmers on a wide scale, it's going to be limited by, what does 120 soil respiration rate really mean to Linus? I know you farm outside, of Oz. So what does that mean to you compared to someone like Justin Salyer that farms outside of Higginsville?
Let me ask you, could you ever flip this, Jeff, and with the data set that you have and the management records you have and the number of years that have been, say, in no-till and cover crop, could you flip this and say, if I had this many years of no-till and this many years of cover crop, could you predict what the biological activity is rather than have to go out there and management? Can you turn that around and not have it be something you have to go out and measure all the time? And but if you got a good history of your management on that field, you could say, well, that I can get a pretty good idea what I'm going to be at based upon that record.
Yeah, I mean, that'd be a good approach. I mean, that's kind of the nice thing about this data set is sometimes you can look at it in terms of yield and then you can flip it around and try and look at it in terms of soil health and what's affecting these. We haven't thought about that. I suppose you could. I think there would probably be inherent, I guess, holes in our data set because of, you can get so much variation by what cover crop you're growing or how often you're growing it. But I don't know to what degree of accuracy we could get, but you could look at that and see, a decade of cover crops versus, occasional cover crops. And within this area, how does that affect your different soil health indicators? Yeah, I think a data set could be built to do that, and ours could be a part of that.
Yeah, so like Justin Salyer said, you need to be able to benchmark those soil health tests. So like per your soil type or whatever, that would go into it, of course.
Yeah, and that's the really nice thing about soil fertility tests is the benchmarks are already there to some extent, and they're not quite as inherently restricted or limited like the soil health tests are just by soil characteristics. The other question is, I think going back to Linus's question, that's the follow-up question to our data set is suggesting these soil health indicators are helpful, but management changes require money, cover crops requires, cost. Doing these different conservation practices requires like there are economic costs associated with this. To justify, one way that these costs might be offset, and a lot of soil health proponents are suggesting is, you can decrease your fertilizer application. So our data suggests, some of these soil biology tests can help overcome, a supposed fertilizer deficit, but we don't know if that's what the cost or what the economic benefits of that are. So I think that's also an important part of moving forward.
Well, I think we probably ought to bring this to a close. I think we're not gonna. The only bad thing about this meeting compared to last year is we don't have a good barbecued lunch to go along with it. It just doesn't do it for us. So maybe next year, we'll see.
Yeah, maybe next year.
All
Yeah,
right.
hopefully.